Welcome back, class 10, to the online bio classes. Today we are going to continue with the second chapter, structure of chromosomes, which we had already started in the previous class. You remember that we had studied about the different types of chromosomes based on the position of centromeres, like metacentric, submetacentric, telocentric, and acrocentric. These four types of chromosomes we studied according to the position of centromere. Now today we have to study the chromosome packaging. How the chromosomes are packed inside the nucleus of a cell. Now you know that the length of a typical mammalian chromosome, a typical mammalian cell contains chromosomes whose dimension is 2.2 meter. 2.2 meter, okay? The length of the DNA double helix in a typical mammalian cell, we are mammals only, so in our body, the length of the DNA double helix is 2.2 meter. DNA double helix length in mammalian cells or in mammals. Now, such a long polymer is packed inside a typical nucleus whose dimension is only, it is 10 to the power minus 6 meter. 10 to the power minus 6 meter. 10 to the power minus 6 meter is the length of or the dimensions of a nucleus. Now it is in minus 6, 10 to the power minus 6. In such a small nucleus, such a long polymer, how it is packed? Question is here. How is such a long polymer packed inside a small nucleus? 10 to the power 6 meter of nucleus, length of the nucleus, accommodates 2.2 meter long DNA double helix molecule. So answer is, this is possible by DNA packaging or chromosomal packaging. Okay? So in eukaryotes, the length or the DNA double helix is packed, such a long DNA double helix is packed inside the nucleus of a cell by the process called DNA packaging. Means DNA must be packed. Like whenever we go for any journey, in such a small place, we try to pack many articles which is of our daily needs. Understood? In the same way, in such a small nucleus, we try to pack many chromosomes whose length is far greater than the dimensions of the nucleus itself. Now, the next point is that DNA contains, or the nucleus contains, chromosomes, and chromosomes are made up of chromatin. Now, you may have seen that there is a set of positively charged proteins, positively charged, this charge also you have to remember, proteins present inside the nucleus, positively charged proteins called histones. Histone protein you have heard, which makes the DNA. Positively charged proteins called histones are present in the DNA, and these histone proteins contains a special type of, or these histone proteins are positively charged in nature, whereas the DNA molecule itself is negatively charged. Okay, what is the charge of the DNA? It is negatively charged, whereas the proteins which makes that DNA is positively charged. So these positively charged proteins, they form a structure called nucleosome. Now what is DNA histone octamer? There are 8 molecules of histone proteins which forms histone octamer. Like suppose this is the histone octamer. 8 molecules. 8 molecules of histone forms histone octamer. Octa means 8. Histone octamer. Now, if this structure is of histone octamer, Okay. Around this histone octamer, there is packing of, or there is wrapping of DNA molecules. Like I am trying to show like this. This is the DNA molecule being packed around it. This is DNA which is negatively charged. This one is the histone protein. This one is also histone. Now the histone protein try to wrap around this histone protein. What is the charge? Negatively charged. Positively charged. Around this positively charged histone proteins, the negatively charged DNA molecule wraps, and this forms a structure called nucleosomes. So what is nucleosomes? Nucleosomes are the structure which is formed by the wrapping of DNA around the positively charged histone particles. Okay? Now these histone particles are positively charged and the DNA molecule is negatively charged. On the surface of chromosomes, they are seen as beads on a string. So it is also called as beads on a string. Beads on a string structure. Now what is this bead? Bead means just like I have drawn here, the swellings are there. Okay? And the string means the chromosome or the chromatin on which these nucleosomes are present. They form the repeating unit of, like once it is here, again it is present here, these nucleosomes. So they form the repetitive unit of chromatin. What is chromatin? You know, the thread-like structure which is present inside the nucleus. It is called chromatin. And on this chromatin, these beads on a string-like structure is seen, which are seen under the electron microscope. During metaphase stage of cell division, there will be condensation of this chromatin. Chromatin means the one which can be colored bodies. These are the colored bodies which will stain with a particular dye. And during metaphase stage of cell division, these chromatins will condense and they will coil further to form the chromosomes. Okay? So you can see how starting from histone protein and the histone protein and the DNA molecules, how they arrange themselves to form the structure called nucleosomes. Nucleosomes are the unit at the smallest part of the chromatin, and this chromatin condenses and coils to form the chromosomes. And in this way, there is packaging of DNA molecules inside the nucleus of a cell. Now, there is one more term called non-histone proteins. In higher organisms, the packaging of DNA inside the nucleus requires additional set of proteins, and they are called non-histone chromosomal proteins, NHC. NHC means non-histone chromosomal proteins. So what are they? They are the proteins, histone proteins, which are required for packaging of chromatin material at higher levels. Understood? So what are the terms we studied in chromosome packaging? The question was, the size of a nucleus is very small, 10 to the power minus 6 meter, whereas the length of chromosomes which are present inside the nucleus is, it is almost like this chromosomes is made up of DNA, and the structure of one DNA double helix, this is called DNA double helix structure, it is nearly 2.2 meter. One of the DNA measures 2.2 meter, such a long length, and it is present inside the nucleus whose size is only 10 to the power minus 6 meter, already written here. 2.2 meter of DNA double helix, it fits inside a small place of 10 to the power minus 6 meter, which is the dimensions of a nucleus. And the question was, how is this possible? The answer is by DNA or chromosomal packaging. Now how it happens? That there are positively charged proteins called histone proteins. Okay? And these histone proteins, they form an octamer in which 8 molecules of histone proteins are present. And the histone proteins which are positively charged is wrapped. Wrapped means surrounded by negatively charged DNA molecule or the DNA to form a structure known as nucleosomes. And nucleosomes, they form the repetitive unit on a chromatin. Chromatin means this thread-like structure. They form a repetitive unit. They are also known as bits on a string structure, which is seen with the electron microscope. And this chromatin network, they condenses and coils further during the metaphase stage of cell division to form chromosomes. And apart from these histone proteins, there are some extra non-histone chromosomal proteins, the non-histone chromosomal proteins which are required for packaging of DNA in higher levels. Okay, so now next we will study about the terminology which are associated with this chapter. Okay. Okay, now the next term I told, there are certain terms which we have to learn associated with these chromosomes. The first one is chromatin. Just now I had shown you with the help of a diagram also. Inside the nucleus of a cell, the thread-like structures which are present is called chromatin. This chromatin network, it is seen in the interphase stage of cell division also, in the resting stage. Now what are they made up of? They are mainly made up of proteins called histone proteins and the DNA. So chromatin is made up of two parts, DNA plus protein, which are packed together inside the nucleus of a cell. Now they have three components or three parts. What are they? Number A, the first one is euchromatin. What is euchromatin? Euchromatin is that part of the chromatin which stain lightly, means they can be colored with the light dyes, and they are transcriptionally, means transcription is the process by which the replication of DNA occurs. So euchromatin is transcriptionally very active and it is lightly stained. So if you have to write the definition, you will say that DNA wraps around the histone proteins forming nucleosomes. I had already said about it that DNA coils around the histone proteins to form nucleosomes. The unit is called nucleosomes. They appear like bits on a string structure. Like this is the string and they are the bits. So they give the appearance of bits on a string structure, and it stains lightly with the stains. It can be colored with the light stains. It will take light color, not the deep color, and that's why it is also known as euchromatin. And they are transcriptionally active. So active in transcription, that point we can write. And they stain light. They appear like bits on a string. That also we can add. Bits on a string like appearance is there in euchromatin. Now second one is heterochromatin. Heterochromatin, that part of the chromatin which has a length of 30 nm. The region it wraps into 30 nm fiber. The length of the fiber is 30 nm. Fiber is formed by the wrapping of this chromatin network, and they stain darkly. So what is the definition you have to write? DNA wraps around the histone protein, that multiple histones wrap. Here only one histone is there, like 8 molecules. There will be multiple histones, means many histone proteins will be there. They will wrap into a 30 nm fiber which consists of the nucleosomes in the compact form. Like nucleosomes will be very tightly packed because many histones are present, and that these regions stain darkly and they are transcriptionally inactive. So inactive transcriptionally, in transcription means they are not helping in the process of DNA replication, and they stain darkly. This one is staying light in color, this one stays dark in color. Okay. Now further if we go, that is, if we make higher level DNA packaging in which there is more than 30 nm fiber, if the length of the fiber increases to more than 30, then it happens in the metaphase chromosome during mitosis and meiosis. Now next point is number 2. The second term that we have to do is chromatid. First one was chromatin, second one is chromatid. What is chromatid? You may have seen that if there are, this is one chromosome which consists of two arms. There are also two arms I am drawing, and both of them are attached in the middle with the help of a point called centromere. So till both the arms are attached with the centromere, it is called a chromosome or a chromatid. But once they separate from it, we say that they are chromosomes. Till they are joined, they are known as chromosomes. But once they separate, they become chromatid. So what is chromatid? In simple words that we can say, chromatid is the part of chromosome. So what is the definition given in your book also? A chromatid is one copy of a duplicated chromosome. I'll just write it here. What is chromatid? A copy of chromosome. Duplicated means the chromosome which has undergone division to form two copies. So what is chromatid? Okay, this is one chromatid, this is another chromatid. Two chromatids are here. So these are also known as chromatid. If I draw it separately, this one is one chromatid, a single chromatid. Since duplication occurs, there are two chromatids, now it forms a chromosome. This entire structure is called chromosomes. The one which is there in the center, this one, it is known as centromere. The point of contact between the two chromatids is known as centromere. And at the time of cell division, there is separation, like these two chromatids will get separated during anaphase stage of mitosis, cell division, as well as during the meiosis cell division. There will be separation. Even the centromere will also divide. Like there is one centromere, now two centromeres will be there, and both of them will get separated, and they will start moving towards opposite poles. Now they will be called as chromosomes only. Daughter chromosomes we can say, or chromatid also we can say. Earlier also they called as chromosome. Even after separation from each other, when they move towards opposite poles during anaphase stage, we can call them as daughter chromatids or daughter chromosomes or as chromatid. So what is chromatid? It is a part of chromosome we can say. Okay, there are two chromatids, and both the chromatids of the same chromosome, these two, they are also known as sister chromatids. Here also we can write, they are called sister chromatids. But if I draw two different chromosomes, like this one is separate, this one is separate, then they are called non-sister chromatids. Got it? Now next we have the word called gene, the third point. What is gene? Gene is the structural and functional unit of chromosomes. What is gene? It is a fragment of DNA which constitute the chromosomes is called gene, and it contains information, all the genetic information of an organism. Every individual carry one pair of genes. So like one pair of gene is there, capital T, small t. This is the gene for height, in which this one is the character of tallness, and this small t represents dwarfness. So what is gene? How to write the definition? Genes are the functional part of DNA. So you can write genes are functional part of DNA. And you know just now what is DNA. DNA stands for deoxyribonucleic acid, the one which makes our chromosomes. Okay. So out of one pair, every individual gene has one pair of, like every gene has two forms. Just now I said that every individual, every person has two copies of each gene. So one gene has two copies. One of them comes from father, then the other one comes from mother. Okay, this is called paternal, this one is called maternal, and they contribute towards the characters of an organism. Alright, so now next we will study about the structure of DNA and who gave this model and what is the diagram of DNA.